Well, thank you very much for the opportunity to speak here today. It's actually great to see so many colleagues here in the audience. I'd like to spend today telling you about some of the efforts that we've had in brain mapping, in particular brain mapping at a cellular level, but then also how we can use this map of cell types across the brain, both to understand disease and potentially to treat disease in precision medicine types of approaches. I have no disclosures to make. So, technology drives so much of what we do in science. And until quite recently, we've had really not very many tools to study the human brain itself. It's extremely inaccessible, it's very large, it's complex, it's difficult to get high quality tissues. But that's really changed in the last 5 or so years, where we now have tools to allow us to classify the types of cells in the brain, characterize those cells, and then target and manipulate those cells. The classification has actually come from the field of genomics. Single-cell genomics methods can be applied to post-mortem brains and really start to plumb the depths of the diversity of cell types, as I'll describe in a bit more detail. We have other tools at our disposal to begin to characterize the properties of those brains. And in particular, at least in certain parts of the brain, we can get access to living neurosurgical resections and apply techniques like slice electrophysiology or modern instantiations of this like PAP-seq to begin to characterize these. And then finally, a molecular approach to understanding the types of cells that make up the nervous system also gives us insight into regulatory biology and the identification of regulatory sequences that can be used to drive selective manipulation or potentially selective treatment. So I'm going to spend the first part of this talk giving you a really high-level overview of some of these cell atlas studies. These are large-scale consortium efforts, as you mentioned, and there are many here at UW that are involved with these efforts as well. And then 2 examples of how we can use these. One to understand vulnerable cell populations in Alzheimer's disease, and another to use cell-selective approaches to therapeutics in the case of genetic epilepsies. So the real key technology that's changed this field are the single-cell genomics methods. And by that I mean single-cell RNA sequencing, and now single-cell epigenetic methods as well. These are extremely high information content methods. You can run sequencing on now millions of cells. You can measure ostensibly all of the genes that are actively being used in those cells. And that molecular fingerprint then defines the cell. And so you can use this to develop a classification of types of cells based on their molecular signatures. This works extremely well, so I'll demonstrate soon. And we're able to build taxonomies of cells that sort of form a framework for understanding the system. And now we can begin to layer on information. We can begin to characterize those cells in a whole variety of different ways. A lot of this work has been sponsored by the NIH BRAIN Initiative Cell Census Project. This is a very large-scale consortium effort intended to bring together a wide variety of expertise for a coordinated project to map the brain. The BRAIN Initiative Cell Census Network was the initial phase of this, BICCN, and at that point, the technologies had become robust enough to think about doing an entire model system, an entire mouse brain, and try to systematically map this brain. At the same time, this effort was intended to scale the methods to be able to approach larger brains and more complex brains, most notably human. That was so successful that it led to the next phase of this, which is called the BRAIN Initiative Cell Atlas Network, or BICAN, that we're about halfway through now, which is intended to really move to a comprehensive atlas of the human brain. So, but let me describe what these atlases look like. The first instantiation of this in BICCN was to focus on one part of the cortex, the primary motor cortex, and to really test this idea of being able to create a comprehensive quantitative map of the cell types that make up the system. And that kind of defined the paradigm for what we mean by a brain cell atlas. It is a transcriptionally based classification of cells that can be described hierarchically. This is just what the biology tells us here, that you can break out big groups of the neurons versus the non-neuronal cells. If we follow the neuronal lineages, you get the excitatory neurons versus the inhibitory neurons that come from different developmental zones. Keep going down levels, for example, to the parvalbumin positive, somatostatin positive, VIP positive, GABAergic interneurons, and another level deeper than that as well. And so it sort of recapitulates basically everything that we knew from the field about the types of cells and their origins, plus another level of resolution that we didn't have before, and the full molecular profile that gives rise to the properties of those cells. So it's a description of the cell types, it's their expression profiles, their epigenetic profiles, their spatial distributions in the tissue, and the properties that begin to get layered on top of that. And in particular, we can start to bring in cellular morphology, cellular physiology, connectivity, for example. And it also turns out that you can do this across species. So you can do this in any species. And this information is enough, there's enough conservation of the molecular signatures to be able to quantitatively align homologous types across species. And so you end up with this highly integrated, quantitatively mapped cross-species atlas where a lot of things are actually very well conserved, but you can also find things that are highly specialized in individual species. I wanted to mention one other thing in particular. In the mouse, of course, there's a lot that one can do. In the human, we're a bit more limited. But we nevertheless can really do a lot. We can do this molecular profiling, we can now do the spatial mapping, and we can characterize the properties of cells where we get access to tissues. And we've had a very fruitful, long-standing collaboration with a variety of neurosurgeons in the Department of Neurological Surgery here, where we get access to tissue resections from either tumor resections on the periphery or epilepsy treatment, where you have to core through the middle temporal gyrus typically. And we can use those tissues, they're very healthy, we can characterize the properties of cells and map them to this molecular taxonomy. So you end up with this highly annotated, curated reference, and that's sort of the point of what I'm trying to get across in this first thing, that we get a reference out of this. In just a few years after that first region, this already moved to whole brain coverage. So in the mouse, we now have a whole brain atlas of both the typology and also the spatial distributions of those cells. And it turns out to be extraordinarily complex. There are more than 5,000 discriminable types of cells in the brain. That's probably 2 orders of magnitude more than most other organ systems. So we really have an intensely complicated system here, but we can define it now. In parallel with that, we came up with the first draft of a human brain atlas that already characterized about 3,000 types of cells in this phase. This has now led to the next phase, and we're currently running one of the projects in this BICAN consortium, which is called the Human and Mammalian Brain Atlas Project, or HMBA. And this is really an effort to see if we can scale this up to do the entire human brain comprehensively with both single cell and spatial methods, and link it to the neuroimaging field as well. And so it's really a consortium project that, fortunately, because of the BRAIN Initiative, we were able to assemble the right group of people to tackle that. And just to give you a little bit more detail of where that's going, this is now intended to be an atlas that covers the entire human brain, the most commonly used biomedically relevant model organisms, so macaque and marmoset and mouse, and to align these all across species to get complete brain-wide coverage, single cell and spatial, linking into the imaging space and begin to layer on information and properties about those cells. And the real sort of philosophy behind this is that we're trying to create something similar to the genome. This is a foundational reference. We all see the same thing when we measure these. Let's start to standardize this and make it a highly annotated resource of the sort that the genome has been for genomics, where we can have now formal releases, we can have standardized nomenclatures around these, we can have tools to visualize and mine and access the data. We can have tools to let you map your own data against it and transfer labels onto it, similar to BLAST for genomics, and then knowledge bases to begin to aggregate this information. And so we're really well on our way to creating this type of reference. And I'm not going to describe the science around this, but rather the applications for the remainder of this talk. So the first thing that we can do with this is to use this knowledge base now to help to understand disease phenotypes. And in particular, I'll discuss a project called the Seattle Alzheimer's Disease Brain Cell Atlas, or CAD project. This is a really incredible collaboration between the Allen Institute, University of Washington, and Kaiser Permanente to try to apply these tools now on a cohort scale to begin to understand disease phenotypes at this very fine cellular level that we can gain from using this atlas. This has been a terrific collaboration with several colleagues in particular I'd like to call out. Dirk Keen, Tom Grabowski, Eric Larson is in the audience here, Paul Crane, and many others here as well, that really bring our complementary strengths together. The aspiration is pretty simple. Can we combine our understanding of the neuropathology driving progression of Alzheimer's disease with these new types of tools and start to understand what kinds of cells are affected, where they're located in the brain, and when they're located along the progression of Alzheimer's? And we can ask all kinds of questions with these kinds of tools that we have now. Are there selectively vulnerable cell populations that are vulnerable at different times in disease? Can we map disease trajectories with these kinds of data and identify molecular pathways that are affected? And can we identify cellular targets that might actually be targets for therapeutics in the long run? Put a slightly different way, we want to bring the resolution of cell and circuit neuroscience that we typically have in model organisms to our understanding of human disease. So to begin with, we have to go to the start. We need to have access to really high quality tissues. And we're very fortunate to work with Dirk Keen and his colleagues here in DLMP, who have adapted methods for preparing pretty optimally human brain postmortem tissues for working with these single cell and spatial methods. Dirk has taken some methods that we initially tried to develop at the Allen Institute and turned them into a full brain banking enterprise to do this. And multiple times now has sort of reinvented how brain preparation is done to improve the quality of the tissues and our ability to have very precise anatomy. For this purpose, really this involves thinner slabbing, optimized freezing of standardized preparation, photo documentation, so that we can really understand these tissues and identify regions very consistently. And with this type of preparation, we actually were able to just use a consecutive set of brains that came into his clinic rather than having to cherry pick ones that had high quality. They essentially all have high enough quality to work with. This project was very much a collaboration with both the ADRC here at UW as well as the Adult Changes in Thought, or ACT program at Kaiser that Eric has been involved in for many, many years. And the two of these really created a good cohort for this study to approach, because we wanted to have a cohort that spanned the spectrum of AD pathology. And so in the case of the ADRC, most people come in, they're already symptomatic. By having a population-based cohort, we're also able to get individuals across the full spectrum from no pathology to very high pathology. Together, we were able to assemble a cohort that met these criteria. This initial study had 84 individuals in it, which is actually a large number for single-cell genomics studies, or it was at the time because it's very expensive to do these studies. And this really spanned the spectrum of pathology from the perspective of Brock staging, of Thal staging, SERAD, and about half of these cases were pure AD pathology, about half actually had other comorbidities as well. But this presented a cohort that really does span the spectrum of pathology from none to very high to allow us to think about building trajectories. This allows us now to think about two different major kinds of analysis that we could do. One is to take a brain region and look at increasing severity, by which I mean increasing pathology. And the other is to look at regional progression with these same methods. And obviously a single region is easier than multiple regions, so we started with one, but we now have moved to many regions, and I'll try to describe results from both of these approaches. But again, the idea is let's follow the pathology. The approach to this was to bring together the expertise from both sides here, so to bring quantitative neuropathology that we can do on these brain tissues and marry it with single-cell genomics and spatial genomics that we can do on the same brains in adjacent tissues to really map these things together. On the first side, this involved bringing machine learning for signal quantification and feature extraction on image-based neuropathology. So this involves many of the usual culprits, A-beta, hyperphosphorylated tau. We also brought in a panel, so we have TDP43, alpha-synuclein, and a series of markers for different cells, such as NUN for neurons, which turned out not to be a stable marker, GFAP for astrocytes, IBO1 for microglia, and to try to build a suite of metrics that we could then map a progression of disease. Using those information, we can then build bioinformatically or biophysically inspired models to map this quantitative neuropathology from a seeding of pathology to exponential rise. This builds an ordering of the individuals along a pseudo-progression that, as you can see here, really maps very well to increases in the amyloid and tau pathology. It also maps very well to independent measures that weren't part of this, like cognitive scores, ADNC, Thal and Brock. And then if we look at this, at the nature of this trajectory a bit more carefully, it really seems to break this increasing severity of the pathology into two phases: an early epoch where it's largely linear aggregation and preclinical, versus a later epoch where it starts to become much more exponential and much more associated with cognitive decline. So I wanted to mention this because some of the effects that I'll show later are more associated with this earlier phase when there still isn't a lot of pathology. Now to begin to ask questions about the cell types, we performed either RNA sequencing or multi-omics on all of these individuals and then mapped them into the reference. And this is actually kind of interesting because the reference is actually built on only a few individuals, and now we're doing this study of a cohort, and so we have much more data to put into this. For the most part, the reference held up, but actually it did end up embellishing the reference and much better coverage of non-neuronal cells, for example, as well as a few disease-associated states. There are states of microglia and astrocytes, which are really only seen in 80 cases. So we end up with, at the end of the day, a reference that's about 139 types of cells in just one single region of the cortex that we can now begin to ask questions about. And I should also say that essentially all of the individuals have representation of all of these types of cells. This is a very robust and consistent sort of blueprint of the human brain that you see even in individuals that may be over 100 and have high pathology, they still have these cells. One last thing before I get to the actual results. So this was for one region. Doing this across a series of regions and being able to ask coherent questions made us create a taxonomy that's now integrated across all of those different regions, so we could be looking at similar cell types. And so now instead of a single region, we have 10 regions that we have to map together. Fortunately, cortical regions have a pretty stereotyped blueprint, and you can do this for at least down to a certain level. And so this is just sort of illustrating that we can integrate data from various regions, and for the most part they align. There are some regional differences. For example, in the hippocampus, the excitatory neurons are quite different. And for example, primary visual cortex has some specialized cell types as well. But for the most part, we can get this coherent classification that now we can ask all kinds of questions for. So I'm going to describe some pretty complicated results in the next couple slides. So bear with me, I'm going to try to sort of unpack this a little bit. But the first thing that we can ask is whether there are vulnerable cell populations or affected cell populations which can be seen with changes in abundance of those cells. So in the case of neurons, we would expect them to be lost. In the case of certain disease states, we might expect them to go up. And so here I'm illustrating for the first region we looked at, which is the middle temporal gyrus, the relative changes across AD progression that you can see. If I could focus your attention initially on the right-hand side here, you see several types of cells that actually go up in proportion. And these are the well-described disease-associated microglia and astrocytes. So we do in fact see some cell populations that increase in their relative proportion. They are exclusively non-neuronal populations. If you look over on the left, on the other hand, these are the neuronal populations, and I just boxed here the excitatory neuronal populations. And I think you can see at a glance that most of these cell populations are actually not affected. But there are several types that are really dramatically affected, and in particular, the layer 2-3 excitatory neurons in the upper layers of the cortex show substantial decreases as a function of increased pathology. It's not just the excitatory cells. On the left-hand side here, there also are a variety of interneuron types that are also affected, of the somatostatin, parvalbumin, VIP, SNCG, and LAMP5 types all have affected types, but it's not all of them. There's selectivity in which types are affected there as well. So now let me take you to look at a cross-regional effort here as well. So now we've gone from the middle temporal gyrus, which is actually sort of in the middle of this progression, and included now earlier affected regions, hippocampus moving into the entorhinal cortex, moving up to ITG and into MTG, and then after MTG as well into STG, frontal cortex, V1, angular gyrus, for example. And so if I start here again from the MTG, now I'm going to move backwards. This is moving into the inferior temporal gyrus, moving into the entorhinal cortex, medial, lateral, and hippocampus. And what I want to draw your attention to is how incredibly similar this pattern is across cortical areas. It's really remarkable how the same types of cells seem to be vulnerable in the different areas, but we haven't gotten to time yet. And we move in the other direction, moving to STG, frontal insular cortex, dorsolateral prefrontal cortex, angular gyrus, and primary visual cortex. Even in primary visual cortex, it's still fundamentally the same types of cells that are affected. So this seems to be a sort of a canonical vulnerability to pathology that we're seeing across areas. I will point out that there are some regional specific vulnerabilities as well. And one in particular I'll call out here, primary visual cortex is, as anyone who's looked at histology of V1 knows, is a very different organization of cortex. We see a different cellular organization as well. There are specialized cell populations. The most notable are the layer 4 input cells that are receiving thalamic input are dramatically expanded in V1. They turn out to be transcriptomically very different from those in other areas as well. And they turn out to be very vulnerable, which was a bit of a surprise because really V1 is typically thought as a bit more spared area. But in fact, there is a literature that suggests all sorts of visual deficits in Alzheimer's. And we might have a substrate for this in actually the input layer is being affected, which would almost certainly affect anything downstream from that. So I think we're starting to get at a pretty good understanding of which types of cells are affected, at least in these cortical regions. And I think a big question in the field, because a lot of people like to use model systems, is whether such a thing is actually recapitulated in mouse models. And we've done our best job to try to take available data in the field from different mouse models and do these same types of analyses. And we do not see the same thing. So we see very strongly a microglial phenotype. There's a disease-associated state that you can see extremely clearly in many different studies. But this selective neuronal vulnerability does not seem to be a feature of most of these mice. So that's important to know, since a lot of effort's going into these mice, but hopefully now we have something of a guide to help to improve models over time. So now let me move to the timing of events. So so far I've just showed you what types of cells are affected, but not when they're affected. And so if we begin to now use the pseudo-trajectory to look at what's affected when, we can see, for example, that these disease-associated non-neuronal phenotypes start early and persist through the increasing severity of pathology. On the other hand, if we look at neurons, things are happening at different times. And the thing that, of course, is very well known in the field is the loss of cortical projection neurons, and in particular the upper layer ones. And you can see that these are indeed affected, but they're affected very late in this progression, well into this sort of exponential phase of the pathology that we described. In contrast, the somatostatin neurons are actually affected very early, well before there's substantial pathology. So that was actually quite a surprise. I mean, other people have described somatostatin, but not in the context of it happening earlier. And so we've been looking now to see whether this is a conserved phenotype across areas. Here is showing you dorsolateral prefrontal cortex. It looks the same, and it really looks the same across all the cortical areas. So this seems to be an extremely early hallmark of disease, and a very specific one. We could take this another step further and look to see which types of somatostatin cells and where they're located in the cortex. To do this, we bring the technique of MERFISH to do a highly multiplexed gene panel with a set of markers that collectively can discriminate among all these types of cells and map their spatial distributions. It turns out that the vulnerable somatostatin populations are in the upper layers of the cortex, and the deeper ones are not vulnerable in this sense. And interestingly as well, that places them intermingled with all of the other major vulnerable populations. So the layer 2-3 excitatory cells, the VIP neurons, for example, are predominantly located in upper layers. So most of the vulnerable cell populations are co-localized, and so you can begin to think that maybe there's a chain of events where the loss of one might lead to loss of the others. And then finally, the reason I mentioned the neurosurgical connection at the beginning is that we have information about the phenotypes of these cells as well, and we know what these somatostatin cells now look like. Somatostatin neurons are well known to be very diverse. There are many morphological types of interneurons. And furthermore, some of these are species specialized. And so one of the major groups of these somatostatin cells in the human are called double bouquet cells for their shape. And these are not found in rodents either. So interestingly, we have an affected cell population that's not, the vulnerability overall is not conserved in the mouse models. And this type of cell isn't even found in the mouse models. So it really creates a challenge of how we're going to study this effectively. But on the positive side, we now have targets and we have some new ideas that we can think about. And as we started to get these results, really began to change my perspective of what's happening in Alzheimer's. That, if you think of the therapeutic, the industry and what they're focused on, it's a proteinopathy. If you come from the genetic side, it's microglia that are really the target. But of course, that's not going to lead to cognitive decline. This is going to be neural circuit issues. And as we start to see the very specific types of cells are affected circuit elements in sequence, maybe we should be thinking of this more as a circuit disease, actually, and what the consequences of that would be. Somatostatin cells have a really powerful effect on controlling cortical circuitry. They inhibit all other types of cells except for themselves. You can imagine a pretty significant disruption of excitatory inhibitory balance as a consequence of the loss of these, and there are hyperexcitability phenotypes in early stages of Alzheimer's. They're involved in oscillatory behavior, and you can imagine also that they could be sort of some trophic interactions where you may lose other cells as a consequence of losing these cells, and maybe that's part of a sequence of events. At any rate, it's a series of new ideas. We have new targets and can think about some new ways of thinking about the disease and studying the disease. And the final thing I'll just say is that it also presents a cellular target where maybe we can actually have that as a therapeutic target to go for the types of cells that are vulnerable as opposed to a molecular pathway. Let me just close this part of the talk by saying that I've just chosen to show you one little part of this. It's the incredibly rich data that comes from these omics analyses. We have profiles of all 139 types across all these different regions now, and understand a lot about the properties of these cells. I haven't talked at all about molecular pathways that are affected, but there are molecular pathways affected in all types of cells, even if they're not vulnerable to the point of being lost. So this is a rich resource to look at, and we've tried to create an accessible resource for the community to have access to these data and mine it to their own interests. So I want to finish this talk on moving from description to perturbation, or to being able to do something with these kinds of data. If we can begin to identify vulnerable cell populations, we can think about targeting them, and we now have the tools to be able to do this. An interesting outcome that I think was not fully anticipated when we began all of this atlasing work is that we would be able not only to describe these cell populations, but identify regulatory sequences that are responsible for the cell type specificity of their expression. And it turns out that not the RNA-seq data, but the attack-seq data is really gold for being able to identify regulatory regions in a way that's been frustrating for the field for many decades, where you go promoter bashing. The regulatory regions tend to be open chromatin, and the attack-seq method can systematically sequence all the regions that are open genome-wide in individual cells, just like you can for RNA-seq, and then you can mine for cell-type selective peaks that may be enhancers. They turn out to be very short, typically on the order of 500 nucleotides or so, which is great because it fits into a viral vector as a tool to test. We can clone these into AAVs as a testing mechanism. We can couple this with advances in capsids for things like blood-brain barrier crossing, which makes this a very easy experiment. And you can have an expression cassette that's now delivered to infect essentially all cells in a brain. And now you can see what the specificity conferred by that potential regulatory element is. We can do this in mouse, we can do it in monkey. So typically we would end up with sort of a screen where you mine for these, you clone them, you screen them, and then things that pass in mouse, maybe we take to monkey to see that this might be something that could be useful for therapeutic purposes. And just to give you a couple examples here, here in the mouse, on the left-hand side here is a mouse with a pan-neuronal enhancer, or promoter in this case. And you can basically see that every cell, every neuron is infected and expressing. With a GABAergic specific one, just forebrain GABAergic neurons, you can really restrict that. We can get into extraordinarily specific things, like here's one that only expresses in the CA2 pyramidal neurons. All the neurons were affected, it's just where it got turned on. So fast forward, in the last few years, we've figured out how to really harness this approach and to use it to systematically identify tools that can target circuit elements in different parts of the brain. And to just give you one example here, here is a series of enhancers developed to target the main components of the basal ganglia, the striatum. So for example, we can target the medium spiny neurons. These are the predominant projection GABAergic neurons in the striatum. We can target all of them, have a pan-MSN marker, or just the direct D1 pathway, or just the indirect D2 pathway. We could target all of the individual interneuron types within this, the cholinergic neurons, the chortle neurons, as well as the dopaminergic neurons in the substantia nigra. And so this is sort of where we are now. We can actually take any part of the brain and systematically deconstruct it, understand its types, and develop tools to target those. And those tools work not just in a mouse. We're not limited to mouse anymore. They work across species. And so most of these enhancers, at least at this level of resolution, work and have conserved specificity from a mouse to a rat to a monkey. And in some cases, we can apply these to slices of living human brain from surgical resections and demonstrate that they work in human as well. This is a series of these striatal enhancers that were tested in mouse and in rat. In each case, they have a conserved pattern, and several that were then tested also in macaque monkeys here at the National Primate Research Center, and also have conserved specificity. So the remaining piece to really making this an effective thing is a capsid that works in primates that can go brain-wide, and that is actually happening right now as well. And so we're starting to get the tools to do this sort of primate or potentially human global delivery, local specificity from these regulatory elements. And I just wanted to mention that we had a publication package coming from the Allen Institute and from a larger program, NIH, called the Armamentarium. It's part of the BRAIN Initiative. And we have a genetic tools atlas resource, if any of these are interesting to you in the audience here as well. So then finally, as we started to get this specificity, it became pretty clear that this could be an avenue for precision medicine, where you target the right cell populations. And so we began to explore this in epilepsy, and genetic epilepsies in particular, where there are some well-known targets, and we know the cell populations to target. And so it's an attractive place to start exploring these tools and see whether they might be viable therapeutics. And the first story, I'm going to tell you two stories here in the epilepsy space. The first is in Dravet syndrome, which is a childhood epilepsy. This is a haploinsufficiency of an ion channel called SCN1A. It has a very severe infant mortality, as well as seizure phenotypes. And right now, they're really lacking effective treatments for this. One challenge in approaching this has been that the gene is too long for conventional AAV-based gene therapy, and that's really hampered the field making progress on this. But we decided to tackle this challenge. And One additional piece of information here is that it's known that a lot of the phenotypes of Dravet are due to inhibitory neuron dysfunction. And so if we could target the interneurons, potentially we could have a rescue of this. One of our first really effective enhancers is a DLX enhancer that targets forebrain GABAergic interneurons. This works across species. It even works in a human slice. If you apply this to a human slice. And so we have the ability to deliver to the right cells. And we came up with a method for being able to have a two-part system with a split intine where they're delivered independently, but they can reconstitute a functional channel if they both get in the same cell. So this works. We're able to reconstitute functional SCN1A. And when we then apply this to a mouse model of Dravet, we've done this with several mouse models of Dravet, we can get some really pretty remarkable rescues. So the first thing is that these mice have an infant mortality phenotype as well. About half of the mice will die in the first few weeks after birth. If we deliver this reconstituted SCN1A to the inhibitory neurons, we can get 100% rescue of this mortality phenotype. And we've now kept these mice alive for over a year, and that is maintained over their lives. We can get a very strong rescue of their seizure phenotypes. These have thermally induced seizures that can be induced. We can rescue that. They have spontaneous epileptic symptoms, including interictal spikes, myoclonic, and generalized tonic-clonic that can be really substantially rescued with this approach. And so it seems to rescue most of the dominant phenotypes in a mouse model. But the key thing that I really want to highlight here is that cell type specificity matters for this. And if we deliver this to all neurons instead of the inhibitory neurons, it dramatically worsens the mortality phenotype. So it has no rescue at all at the doses where we can start to get phenotypic rescue on the seizure side of things. So I think that this is going to be the case in many diseases. And if you think of the immune system, we wouldn't target just the immune system. You target certain populations in the immune system. We should start to think about that for brain as well. So I'll give you another example to try to convince you even more. So another genetic epilepsy is SLC6A1 related disorder. This is a GABA transporter. This is also a rare developmental epilepsy due to haploinsufficiency of this gene. It's a little less severe than Dravet, but it has early onset epilepsy, typically absence seizures, and there really are no disease-modifying treatments available right now. Sort of following the train of logic here, we know a lot about the expression of this gene normally. It's expressed in GABAergic cells as well as non-neuronal cell populations, astrocytes and oligodendrocytes. It is a GABA transporter, and so really dysfunction of that is going to inhibit or lead to dysregulation of the amount of GABA around the synapse. So again, similar strategy. We're reintroducing a functional copy of this gene selectively in the inhibitory cells. And here we also can get rescues of many different phenotypes. The absence seizures are essentially removed with this treatment. We can rescue body tremors. We can rescue certain elements of short-term memory that are affected. One detail that I didn't really show on this slide is that we actually needed to go brain-wide inhibitory neuron in this one. Forebrain was not sufficient, so we had to hit them all. And similar to the Dravet, if we go non-selective with this delivery, it turns out to be toxic. So at a high dose where we can rescue seizures, this is actually fully toxic. At middle doses, we get convulsive seizures, and we don't rescue at low doses. So it's just totally ineffective if we do this in the wrong cell populations. So we actually have other examples now as well, but we have this new toolkit here now. It's kind of amazing that we can target just about any kind of cell in the brain or the body, for that matter, or combine them. And so it opens a wide avenue of potential therapeutic opportunities. And I'll just give you a teaser here that this somatostatin population that I mentioned for Alzheimer's disease, we can target as well. So I don't know if this is a viable target, but we can start to at least think in that sort of paradigm that if we can identify the right cells, we can do something with them. So let me just end. I'll say, this brain cell atlasing is not just stamp collecting. This is making atlases for a purpose, and we can really describe the system. We can now use this in any disease. All the things I've told you are generalizable. We can do this for any disease now to get the cellular locus of disease, and then we can target those populations for potential ways to intervene. So with that, I will end, and I just want to thank many, many people. These are large consortium projects. Not really possible to thank everyone here, but I want to thank my many colleagues at the Allen Institute. We really specialize in these large-scale collaborative efforts, and collaborators both on the Brain Initiative side and on the CAD side, and our funding sources from the National Institute on Aging, NIMH, and the Brain Initiative. Thank you very much.
Great, thank you. Questions from the audience? There's one on Zoom here. So it says, when you say brain-wide expression in contrast to forebrain, does this include midbrain, brainstem, and cerebellum? And is the expression uniform as you see it in the cerebral cortex?
Yes, so I do mean all of those structures, and I believe that this approach does cover the GABAergic cells in all of them, but it's possible that it's not completely penetrant. I would have to check exactly. So it's interesting. The GABAergic neurons across the brain are not a homogeneous transcriptomically or developmentally specified population. The forebrain ones are, and so you can find something that targets all of them. So we actually had to take a different approach to get this. We started with a pan-neuronal and added a microRNA binding site that actually restricts it to just the inhibitory cells. So this is sort of how this goes often. Like you have to find the tool to target the population you want. So I think it's mostly pan, but I'm not sure if it's quite 100%.
So maybe to dovetail on that, so what other payloads can you deliver? So presumably you can deliver CRISPR-Cas9, even epigenetic modifiers to modify the activity of the enhancer. Could you comment on what you've tried?
Yeah, so we've started off mostly with the low-hanging fruit of gene augmentation or replacement. But they're completely compatible with any of these other things. So we could do CRISPR modifications, we could do other forms of transcriptional or epigenetic regulators. It's really molecular genetics now. You can put anything in there.
Well, I have many questions, but I'll try to keep moving. First of all, I guess when it comes to the somatostatin subtypes that are susceptible, how do you determine which ones have marked mouse orthologs and which don't? Is that by taking in all the sequencing plus the histology plus what we know from patch-seq? And then which ones do have mouse orthologs, and are they all sort of in that NME nephrosilin expressing ones?
Can you repeat the questions for the Zoom audience, please? They're assuming.
Okay, to summarize, I think, what's the evidence base for these being species specialized? And what do we know about those? Are they the MME positive population? I think you're thinking of the Relin positive ones as well, probably. So the answer to the first question is that when we take all of this transcriptomic data and align across species, it allows us to map homologous cell populations based on their overall transcriptomic patterns. And so we do this with all of the cells from cortex or from this cortical region, and you do these alignments. And then you get alignments at different levels of resolution. The top levels of resolution down to a level that we call the subclass, and subclass would be somatostatin positive, is really unequivocal. Like you can map these across species. We can actually, we've done this, we can map them from an opossum to a human being. So it's a common blueprint. But then things start to get specialized at a more granular level. They get specialized to the point it's not really easy to align them and you're not confident about their alignments. And that's a bit of the problem with these double bouquet cells, that within those somatostatin cells, we're not really sure exactly who corresponds to what. We know their phenotypes from doing this patch-seq. And within species, this is now a straightforward thing. You're just mapping a cell to the same species reference. And so we can annotate the clusters within species. And so we know what those are. We also know that from a long history in the field, as well as work at the Allen Institute in mouse, that there don't seem to be any cells that phenotypically align with these double bouquet cells. So we've kind of got these two pieces of information, but I mean, it may be that they actually are homologous to something that just has some rather different properties, rather than being an ultra-specialized thing, but we're just not confident in being able to map that. So your other question is interesting, because these somatostatin cells express reelin, and they probably are among these ones that have the MME, and with the reelin mutation that rescues a presenilin, now this becomes a very interesting population. So I would view this as a bit speculative that this is a direct link at the moment, but it's pretty interesting that they both affect the ApoE signaling, and you actually have this in same cell populations that seem to be vulnerable. So tantalizing. I'd love to follow up with you more on thinking about what we could do with that, but it could very well be that we're getting some hints that the somatostatin cells are important for other reasons.
That's not fair.
So I understand how open reading frames can define a cell population, but what I don't understand is how you're targeting those specific cells based on the open reading frame. And then I also wanted to know what percentage of cells are targeted when you target it. Are you getting close to 100%?
Well, so we're not really using oh, sure. You wanted some more information about how we're actually doing this targeting. So the first thing is that we are not looking for regulatory sequences that affect a particular gene. We're just looking for regions of open chromatin in the cell population that could potentially be an enhancer. And why is it open? It's probably open because it's being bound by some factor that's in the cell. At the moment, we're still basically black boxing this. So we identify these regions. They can be next to a marker gene. They can be megabases away from anything that seems relevant at all and still be effective. They can also be open in one species and not in another and be effective. And so there aren't really rules to this yet, I would say. But presumably, if these things work across species, they're being bound by factors that are conserved across species. So at any rate, it's not, we're not looking at open reading frames at all. We're just looking for short putative regulatory sequences that turn out to be sufficient in and of themselves when combined with a minimal promoter and an expression cassette.
But how are you targeting it? So you're actually putting something that will bind to the open reading frame in there? So it will only bind to that specific cell type?
No. So we make an expression cassette that's in a virus that has one of these enhancers, has a minimal promoter, and has a reporter gene. And we give a high dose of this virus, inject it into the bloodstream with a capsid that crosses the blood-brain barrier, and it transduces effectively every cell in the brain. And now you've got this expression cassette at loose in a cell. And if the factor is there that normally binds to that, it turns it on. Oh, no, over time, we're trying to understand this, like what factors are doing this. Can we get predictive or even synthetic about our approach to making these? But it's not actually necessary to get useful tools.
Charlie?
So as you look at your pseudo-time trajectories of brains from patients with dementias, are you finding specific cells that have either cell states or cell neighborhoods that are localized, have localized capabilities of repair or regeneration?
That's an interesting question. These are post-mortem brains with a snapshot. So I think we would have to be interpreting the transcriptomic signatures and also getting spatial information about those. I'm not sure we can ask those questions quite yet. Or maybe I'd be happy to follow up with you to think more about how to interpret the data that way. I do think that, you know, we did this to get the dominant trajectory in this one. And I think the next stage is we need much bigger numbers. And we're going to start to see that there's a lot of heterogeneity. There will be, no doubt, multiple trajectories. Subtypes have been defined, both molecular subtypes, topographic subtypes, and cognitive subtypes. And so hopefully we'll start to begin to disentangle this and see whether there are common mechanisms or whether they're distinct ones. But your question is a little deeper than that on what's happening locally around these, and I don't know the answer to that yet.
Kelly?
You mentioned that your analysis of the mouse models of Alzheimer's disease didn't look anything like your human subjects from the single cell study. So I was sort of curious, what are the mouse models modeling? And do you know, from your studies on humans, do you have any insight into maybe a better way to make a mouse model for humans?
Well, the mouse models are perfectly logical in their construction. They try to put high-risk alleles, essentially humanized high-risk alleles into mice and see if they can cause this to recapitulate some of the things that happen. And they do recapitulate some things. They increase amyloid production or hyperphosphorylated tau. They can induce this microglial phenotype, as I was describing very, very clearly. But they don't seem to happen in the same sequence and then progress across the brain in the same way. So, you know, I'm not sure exactly how to make a mouse do that. I do know that John Morrison at UC Davis has made a monkey model where he essentially seeds tau in the entorhinal cortex and then sees that it seems to show that progression across the brain in a similar way to a human. So maybe you need to seed it somewhere to start to get such a progression, whereas if you've just genetically induced this, that's not sufficient.
Clearly, the monogenic epilepsy models were very helpful, right? Even because you can manipulate a single gene and then assess the phenotype and try to rescue it.
Yeah, much simpler.
Alzheimer's is a bigger ask for sure. Other questions? I had
Maybe in the very back.
back then, Ben.
Yeah. I was just wondering if you had enough control brains that you could contrast the Alzheimer's progression with normal aging. Do you see different like cellular changes in like just a normal aging process versus Alzheimer's progression? You might not be powered to detect that with the brains that you have.
Yeah, it's a fabulous question that I'd love to know the answer to. We were not powered in this initial study, in part because the average age in the ACT study is actually quite old. So this was actually an aged study, not an aging study for the most part. On the other hand, we were able to get people that within the same age range that had a wide range of pathologies. So in some sense, age is slightly controlled for. I would expect that some of these things have an aging component, and maybe that's exacerbated by pathology. For example, there is a literature on somatostatin, just measuring somatostatin levels, for example, that reports an aging phenotype there as well. But we see it exacerbated pretty substantially, and now we know the source of where that somatostatin was coming from. So, you know, all of these things, like we need bigger studies. We need larger cohorts that span both, can control for aging and many other demographic factors, and the variety of presentations and co-pathologies as well. And actually, we just got this Alzheimer's project funded for renewal, where we're trying to expand this and explore many of those areas. But it's going to take some time to collect the populations to do that with.
Thanks.
Yep.
You mentioned that you used merfish to confirm which somatostatin positive cells were specifically affected. And then you've also collected all this, various modalities on cell location, cell morphologies, connectivity. Is there a way to more systematically combine this with the single cell RNA sequencing data to more precisely map these cell types? I know you're trying to find this integrated thing to integrate these snRNA-seq signals across the brain, but like, can you combine that with the morphological features to map them? Does that make sense?
Yeah, so, I mean, so right now these are independent measurements. So, for example, the patch-seq is not on 80 cases. We never get living tissue from the 80 cases. So really, but we are able to map it to a molecular type because we do the physiology and at the end you extract the nucleus from the cell, you sequence it. So we can really directly map those things. We don't have access to, we don't have a method right now that will let us do all of that in one tissue. But there are proteomic methods which are coming on now and which we're beginning to explore where you can do highly multiplex proteomics, which really just means a set of antibodies in cleared tissues and with light sheet microscopy, and start to really get an idea of the morphologies of certain kinds of cells to the extent that you have markers for them. We don't yet have markers for these neuronal types. We do for microglia and astrocytes and things like that. So I think we need one more technological innovation to let us put it all together.
Well, thank you, Ed, for a wonderful talk, and thank you all for attending.